Hello and welcome to this core practical video. My name is Mr. Osman, a science teacher at Claremont High School in London. Today I'm going to take you through one of the core practicals required for GCSE Biology. The title of this practical is The Effect of Antibiotics on Microbial Cultures. Your teacher will advise you on the type of antibiotics, antiseptics or plant extract that you will be using for your experiment. To carry out this experiment, you will need the following equipment: Petri dish with lid, screw top bottle of sterile liquid nutrition agar, keep in water bath until needed, bacteria culture in screw top bottle, sterile pipette, sterile spreader in rubber, beaker of disinfectant, 2 small filter paper discs of different antibiotic concentration, small disc of sterile filter paper, sticky tape, marker pen, forceps, ruler, ethanol, Bunsen burner, and heat-resistant mat. I am now going to demonstrate how to successfully pour an agar plate. Keeping the lid on the Petri dish, turn the dish upside down. Use the pen to draw 3 sections on the base. Label one section control and the other 2 sections with the name or concentration of each antibiotic or antiseptic solution. Add your initials and the date near the edge of the dish. Turn the dish right way up. Safely light your Bunsen burner, making sure to keep the valve closed. Remove the liquid nutrition agar from the water bath and dry using a paper towel. Unscrew the cap of the nutrient agar bottle and quickly pass the glass neck of the bottle through the Bunsen flame. Then lift the lid of the Petri dish just enough for the agar to be poured carefully into the dish. Pour enough agar to half fill the depth of the Petri dish base. Replace the dish lid immediately. Leave the agar to solidify. You can also use a pre-prepared agar plate to save some time, or pour the agar plate in pairs. I am now going to demonstrate how to successfully make bacteria plate. Unscrew the cap of the bottle of bacteria culture and quickly pass the neck of the bottle through the Bunsen flame. Draw a small amount of culture into the pipette, then flame the neck of the bottle again and replace the lid. Lifting the lid of the Petri dish as little as possible, gently add a few drops of culture to the agar. Replace the dish lid and place the pipette in a disinfectant. Unwrap the spreader or disinfect, and lifting the dish lid as little as possible, gently spread the culture drops across the agar using a back and forward motion with the spreader. Replace the dish lid and disinfectant the spreader. I am now going to demonstrate how to successfully add antibiotic discs. Sterilize the forceps by dipping them into ethanol, then passing them through the Bunsen flame to ignite the ethanol. Use the forceps to pick up the sterile filter paper disc. Lift the dish lid just enough so that the forceps do not touch the bacterial layer on the agar in the dish, and add carefully the disc on the section labeled control. Replace the dish lid as quickly as possible. Sterilize the forceps again. You are now ready to prepare your antibiotic discs. Use your scissors to remove the specific antibiotics you wish to investigate. Sterilize the forceps by dipping them into ethanol, then passing them through the Bunsen flame to ignite the ethanol. Then repeat the previous step with the 2 of the antibiotic disc. placing it on the appropriate section of agar. Remember to replace the dish lid as quickly as possible. Sterilize the forceps by dipping them into ethanol, then passing them through the Bunsen flame to ignite the ethanol. Tape the lid onto your petri dish with 2 pieces of tape and invert the dish. Leave it at 20 to 25 Celsius for 2 to 3 days. To record your results, you need to look carefully at your dish. Do not open it. Measure the diameter of the circle around each disc where there is no bacterial growth. Calculate the zone of inhibition using an appropriate formula. Use your data on your own knowledge of antiseptics, antibiotics, and plant extracts to explain the result you have obtained during your experiment.